Welcome, everyone, to the first ISV webinar of 2025. Glad you guys could attend. We have a very interesting series being set up for this year. And before I introduce our first speaker of this session, I want to give a little bit of a reminder about other ISV webinar series that we have. So on January 30th, ISV is hosting an industry interaction committee. Is showing a mini symposium on pneumococcal vaccines. You can find the links to that at the ISV website. We also have a next generation vaccinologist webinar series, and in February of 2025, James Allen from the Cleveland Clinic is going to present, along with Michael Sharstad from Icahn School of Medicine in Mount Sinai. One will give a presentation, the other will give a mentoring sessions about setting up academic labs and opportunities for young investigators. And then this webinar series will again appear in February on the 26th with Hulan Shen from Harbin Veterinary Research Institute, and she'll talk about emerging avian infections and vaccines. That webinar will be presented in the afternoon in the Asian time period, and then for Europe and Africa, it'll be in the morning, and then the Western Hemisphere will be presented in the overnight hours. Always remember that all of these webinars are recorded and they are placed on the ISV website on YouTube, so you can always watch them if they're not at a time zone or a time that works for you. For today's presentation, it's my honor, and I'd like to introduce Dr. Tonya Villafana, who is the Vice President, Franchise and Medical Scientific Affairs and Vaccine and Immunotherapies Director at the AstraZeneca. She has spent much of her career developing vaccines and monoclonal antibodies targeting areas for distinct pathogens such as COVID-19, influenza, malaria, Ebola, and RSV, which she'll talk about today, among many others. She was the lead scientist that developed a novel, long-lasting RSV monoclonal antibody, which is the first to offer protection against RSV disease in infants entering their first year. That's what she'll be telling us about today. She's had a long, distinguished career. She received her PhD in immunology from the Weill Cornell University Graduate School of Medicine and a master's in public health from the Harvard School of Public Health. She's well recognized in her field for vaccines and therapeutics. And so I'm very pleased to introduce her today to give the ISV webinar. I'll turn it over to you. Thank you, Dr. Villafana, for being here today.
Thank you, Dr. Ross, and it's a great pleasure to be here today to give the first lecture of 2025. I'm going to go ahead and share my slides. So it's a great pleasure, as I mentioned, to be here today to give this lecture. And I'm going to talk about a program and a product that is near and dear to my heart, which is Nirsevimab, a monoclonal antibody for the prevention of RSV disease in infants. This monoclonal antibody was developed in a collaboration between AstraZeneca and Sanofi, where AstraZeneca led the research and development, manufacturing, and regulatory aspects of the program, and Sanofi is the commercialization lead. Before talking about Nirsevimab specifically, I just wanted to talk a little bit about our unit at AstraZeneca that focuses on vaccines and immune therapies. And we have a long history in delivering innovative interventions for infections to protect people from serious consequences of disease. And on the left-hand side of this slide, you can see really our long experience on highly targeted antibodies, beginning with Synagis as a result of MedImmune, which was an AstraZeneca company, all the way actually most recently to this week, the approval of Cavigale, which is a COVID-19 monoclonal antibody. On the right-hand side, you can see our innovations in vaccine development, namely FluMist, our intranasal vaccine, which recently received approval in the US for home administration, as well as the significant amount of work we did during the COVID pandemic with the development of Vaxzevria, delivering about 3 billion doses of that vaccine which was estimated to save about 6 million lives at the beginning of the pandemic. But we have a rich and clinical pipeline that continues to address not just viral infections, but also bacterial pathogens, with a number of programs currently in development for serious bacterial infections. And you can see on this slide, we have a program for C. diff, which is a very significant problem and has been in the US identified as a significant infection that needs novel interventions. We also have programs in Pseudomonas and staph. And then, of course, our key programs that I mentioned previously in COVID-19, RSV, influenza, but also HMPV, a new virus that actually has been in the press a lot lately as people begin to test more for human metapneumovirus. And we've seen the rising number of infections across the globe, particularly in China over the past winter viral season. So I'll switch now to talk about Nirsevimab and RSV disease. So many of you on the lecture may know about RSV disease. It is really a seasonal virus that typically in the Northern Hemisphere circulates during the winter viral months. It is a very common virus. It's contagious and it infects the respiratory tract, leading to serious consequences for infants and older adults. The infection normally starts as an upper respiratory tract, spreads to the lower respiratory tract, airways, and lung tissues, and results in inflammation, runny nose, sneezing, and congestion. It is the most common cause of bronchiolitis and pneumonia in children less than 1 year old, and it has been associated with increased hospitalization, healthcare resource utilization, recurrent wheeze of early childhood, and maybe long-term consequences as well, including COPD, asthma exacerbations. And in many countries in the world, where the health infrastructure may not be able to cope with the surges that happen during the RSV epidemics, may lead to mortality in infants. It is transmitted by contact with oral and nasal secretions. Just wanted to talk a little bit more about the major subtypes because you'll see this in the presentation as we talk about the target for the monoclonal antibody and also the efficacy data that we present against the subtypes. But there are 2 major subtypes and multiple genotypes of RSV. You can see here it's a pneumovirus. There are 2 surface glycoproteins that are important, the G protein, which mediates the attachment to the host cells, and then the fusion protein, which promotes aggregation of multinucleated cells through fusion. And prefusion F, when you can see the schema of prefusion F on the slide, is the target of vaccines and monoclonal antibodies that have been developed or are currently in development. So prior to infant immunizations being available, and this is a slide that we had developed in 2023, RSV caused a significant burden of disease in US infants. And here you can see where we started from in terms of at the base of the pyramid, what the infections look like in the first year of life with medically attended RSV, lower respiratory tract infections at about 500,000 in the US, significant number of clinic visits and emergency department visits, as well as hospitalizations depending on your RSV season. And what we knew was that 72% of those hospitalizations and 66% of those intensive care unit admissions occurred in healthy and term infants. So not just in infants at risk for the most serious consequences of disease. All infants are at risk for serious disease. And you can see here in the US and in other countries, high-income countries, the number of deaths and mortality related to RSV is low. But in many countries in the world, this is not the same picture where there may be significant mortality related to the disease. In the second year of life, and I'll talk about that because it's relevant to our clinical development program, infants with pre-existing medical conditions such as immunodeficiency, congenital heart disease, or chronic lung disease of prematurity are also associated with increased risk, and therefore need for prevention tools to prevent them from having these serious consequences related to disease. So it was a long road to the development of an effective prevention strategy for all infants for RSV. And this is because of the history of RSV vaccine development. So RSV was discovered in 1956 by Chanock, and the first RSV vaccine trials began with a formalin inactivated virus in 1965. Sadly, during the development of those vaccines, children who were given vaccine prior to a season and then exposed to RSV had enhanced disease. Many children were hospitalized, and in fact, 2 children died in those vaccine trials, and that set the tone for future vaccine development. With a very cautious approach to looking for preventive tools for the infant population. The first proof of principal experiments in animal models for prophylactic antibody administration occurred in the late 70s, early 80s. And we at AstraZeneca and Medimune at the time really believed this was the best approach to move forward for prevention tools in infants to develop antibodies that could be used to protect infants from the disease. So our first product that was developed was RSV IVIG, and that was approved in 1996. And in 1998, Villafana, or Synagis, as many people may be aware, was developed for the prevention of serious RSV disease in the highest risk infants. So these were infants who were premature or had an underlying condition such as congenital heart disease or chronic lung disease of prematurity. And this product was a 15 mg per kg dose given to children through the RSV season monthly, which was about 5 shots during the typical RSV season. In about 2010, the pre-fusion conformation of the protein, of the F protein, was mapped, and this paved the way for many of the studies and many of the products that are in development right now, including Nirsevimab, which is a monoclonal antibody targeting the pre-fusion conformation. And I'll, of course, talk much more about that in detail during the presentation. The pre-fusion F conformation or pre-fusion F protein is also the target for vaccines for maternal immunization. So that was the other approach that was deemed really appropriate for moving forward in infants. So you'll see on this slide, both monoclonal antibodies and maternal immunization are passive immunization approaches to protect the youngest infants who are most vulnerable and have serious consequences related to the disease. Nirsevimab was licensed in the US in 2023, as well as Abraceful, which is the maternal vaccine developed by Pfizer. And we also have another monoclonal antibody that was recently positive results were shared for Merck called Clisrovimab, and that is under regulatory review at this time. So this is a slide from PATH, and PATH is an organization that has quite a bit of experience in development of vaccines in global health. And they track all of the RSV vaccines and monoclonal antibodies that are being developed across the globe, all different modalities, all different platforms. And you can see here on this slide that the different platforms are on the left-hand side, and then the current stage of development for all the different approaches is indicated on the slide. And you can see here the products that have been marketed or approved in RSV, either for pediatric populations or older adult populations, which is the other population that is at significant risk for serious consequences related to RSV disease. So you can always go to the website and get a current view, or the most current view, of what's in development. So when we thought of developing an approach that could be a product that could be used in all infants, we thought about the criteria that we would need for this product, particularly for our monoclonal antibody. And so the criteria are on this slide. We knew that we would need a highly potent antibody from a human donor. We wanted to target a highly conserved epitope on the prefusion F protein, so we selected site 0. And we had the technology to extend the half-life of the antibody to enable it to be used so that one shot would be given to protect for an entire RSV season. And so here you could see the advantages that made this approach the approach that we thought would be the best to develop an immunization for infants. Again, as I mentioned before, it's passive immunization. Monoclonal antibodies have a rapid onset of protection. We designed it so a single intramuscular dose could provide coverage for a typical RSV season of about 5 months. When giving the antibody, we know that well-defined levels of neutralizing antibody, we know exactly how much we're giving and how much would be needed to protect a certain population of infants, and the ability to combine with routine pediatric immunizations was critical as well. And we know from mechanism of action that monoclonal antibodies don't interfere with the use of pediatric vaccines. So Nirsevimab, our monoclonal antibody, had all of these important features that enabled its use for RSV prevention in all infants. And on this slide, to the prefusion conformation of the F protein on the left-hand side of the slide. And you could see that the binding site is at that site 0 epitope, right? Site 0 site. And the epitope is at the upper end of the prefusion F protein. It is a highly potent recombinant IgG1 monoclonal antibody, and we have extended the half-life of the antibody with YTE technology, which enables binding to the neonatal Fc receptor to prolong the half-life. The product is once per season, fixed intramuscular dosing, and dosing is flexible. It can be given at birth or just prior to the RSV season. So on this slide, I'd like to just discuss the pathway to development of Nirsevimab. Our IND for this product was filed in 2014. So development really took about almost a decade to go through all of the clinical development and pull together the package of information that would be submitted to regulators around the globe, as well as to advisory committees. And I'm here really illustrating just a subset of the amount of work that the team did to get this approved and used for babies around the world. The early clinical studies, so those were phase 1 studies in adults before moving into phase 1 studies in infants, were conducted in 2014 and 2015. And in 2016, we began our first pivotal efficacy study in preterm infants, 29 to 35 weeks gestational age. And I'll talk a lot more about that study. And then later on, once the results of that study were received, and we had a positive outcome, we moved into the 2 other pivotal studies, an efficacy study in older gestational age infants called Melody, and then a study in infants at high risk of severe RSV disease similar to the population that was covered by Synergist that I spoke of previously. And that study was called MEDLEY. And then we also conducted a study in immunocompromised children because they were at risk for such serious outcomes related to RSV disease. At the top of the slide, you can see that we had many designations in the US that enabled us to move fairly rapidly through development, but we also had this across the world in Europe as well as in China and Japan. In the US, we had fast track designation and we had breakthrough therapy designation given the high unmet need for a prophylactic product in infants. And our BLA was submitted in 2022 with approvals coming across the globe after submissions in the different regions. We also, over the years, have had multiple interactions in the US, but other parts of the world with technical advisory groups that would recommend pediatric vaccines normally to infants. So in the US, those interactions were with the Advisory Committee on Immunization Practices, because our vision was that this monoclonal antibody, for the first time, going into the broadest population of infants, would be. And so those interactions were key because we needed to pave the pathway to allow the monoclonal antibody to be given like a vaccine in infant immunization programs. Since we have had approval, we continue to see real-world effectiveness data looking at the effectiveness of Nirsevimab beyond our clinical trial development, and I'll talk more about that in subsequent slides as well. So on this slide, I'd like to focus on the two pivotal placebo-controlled efficacy studies to describe Nirsevimab's efficacy that was used for approval in these in the pediatric infant population. And on this slide, you can see the two studies that I mentioned previously. Our phase 2b study, which was conducted in infants with gestational age 29 to 35 weeks, and then our MELODY phase 3 study, which was conducted in infants who were either late preterm or term greater than 35 weeks gestational age. The design of both of these studies was quite similar. Both were 2-to-1 randomization. To one randomization, as I mentioned, in children receiving the intervention versus placebo. The only differences between these programs or these two clinical trials are the following. In the phase 2b study, we only looked at a 50 mg dose, and that dose was selected based on our initial PK modeling and the information that we had from our early development. That was the dose that was moved forward into our phase 2b study. Subsequent to the receiving the results of that phase 2b study, we modified our dosing to optimize the dosing based on weight band. And so 2 doses were taken into our phase 3 study, MELODY, a 50 mg dose for those less than 5 kg and a 100 mg dose for those greater than 5 kg. The other difference in these two clinical studies was that in the MELODY phase 3 study, we followed infants through a second RSV season, and that was done at the request of the regulatory interactions we had to determine that there was no enhanced disease happening when children were exposed to RSV in their second season. Just wanted to comment a little bit more on our dose optimization, moving from our phase 2b study to phase 3. Once we had completed our phase 2b study, a post hoc exposure response analysis indicated that there was a trend to lower efficacy in infants with who were weighed more, and that they had lower Nirsevimab serum exposure. And you can see that illustrated on this slide. And so based on this data, we revised our dosing strategy, as I mentioned on the previous slide, to give those infants who were greater than or equal to 5 kg 100 mg dose so that the exposures would look very similar to those who weighed less than 5 kg. So this strategy really optimizes to ensure that the right dose is given to the right infant. This slide again shows some more details on our phase 2b and MELODY study populations. We've talked about the gestational age of the infants. The second point I'd like to just highlight here is when enrollment occurred. So our phase 2b study was done prior to the COVID-19 pandemic, while our MELODY study, the phase 3 study data that I will show you, occurred partially before COVID-19, and then we had to do a pause in enrollment due to the COVID-19 pandemic, and then resume enrollment to complete the enrollment of the targeted number of infants that we had planned for that study. So, you know, it's interesting because we talk about the pre and post COVID-19 eras of development. And just wanted to highlight what we saw during our development plan. We talked about the randomization and how similar these two studies were, and that the only difference was the optimization of our dosing in phase 3 to have the same exposures for all infants that we studied. In both studies, infants were less than a year of age and entering their first full RSV season. They did not meet AAP or other national guidelines to receive palivizumab, which is the standard of care for those higher risk infants. That would have been in the phase 2b study. And we included more than 136 sites in the Northern Hemisphere and more than 30 sites around the world in the Southern Hemisphere. The data I'm going to show in this presentation today is a pooled efficacy analysis for both the phase 2b and MELODY study populations. And the reason that we can pool this data to look at efficacy are illustrated on the slide. The two infant populations are quite similar in respect to mechanism of action, because the mechanism of action of the product is independent of the gestational age of the infant at birth. I also mentioned that the study designs were similar. For the pooling, we included the phase 2b recipients who received the approved dose, so the right exposure. So those would be the infants less than 5 kg in that study. And we included all infants in the MELODY full enrollment cohort, the 3,000 infants we intended to enroll, and all of whom received the optimal dose that was later approved. So on this slide, I want to highlight the demographics and baseline characteristics between the treatment arms of both studies. And you can see here the amount of infants from each of the study at the very top, each of the studies contributing to the analysis. But the rest of the slide shows that we are quite balanced between groups for all of the parameters listed on the slide. And so this is really important to ensure the robustness of our analysis. Wanted to highlight the case definition for what would constitute a medically attended RSV LRTI case in our clinical study. And this was the primary efficacy endpoint in both the phase 2b and the phase 3 MELODY study. And in fact, this is the case definition used for all our studies in the program. So in order to qualify as a case in the study, the infant had to be positive for RSV, and this was determined by a central laboratory RT-PCR assay. And they had to have a sign of lower tract involvement, and that is indicated on the slide. So at least one sign of any of the following indications or lower indications of lower respiratory tract involvement in the middle column. And they also had to have a sign of severity. So at least one sign of any of the indications of severity in the third column on the slide. Medically attended RSV LRTI hospitalization, which you will see in the efficacy results, corresponds to infants who met that primary case definition but were hospitalized. And medically attended RSV LRTI very severe corresponds to those infants who were hospitalized and required supplemental oxygen or IV fluids. So this slide illustrates the robust efficacy seen in the program. And here you can see the LRTIs of different severities and also RSV subtypes, as I had mentioned earlier. So overall, medically attended RSV LRTI efficacy in the pooled population was 79%. And then you can see that efficacy was similarly high, both against subtype A and subtype B in the pooled analysis. Medically attended RSV LRTI hospitalization was also 80.6% and also consistently high for efficacy against severe RSV LRTI. Again, those infants requiring oxygen or supplemental fluids. We also looked at those infants who required ICU admission, and the efficacy is illustrated on the slide, as well as those infants requiring any respiratory support. So the important takeaway from this slide is really the high efficacy that we've seen with the product across infants of varying gestational ages, high efficacy, but consistent efficacy across the endpoints that we studied. This is just some more detail on efficacy because, of course, we wanted to look at it across a number of different parameters. And it's very important here to just, again, show that efficacy was consistently high across hemispheres, across different gestational age infants. In this third one, it's a wide interval because of the lower number of infants. For males and females, efficacy was high across different ethnicities, the different weights, and of course, in different regions. So in conclusion, with efficacy, the pooled analysis demonstrated a 79% efficacy against the primary case definition in infants greater than or equal to 29 weeks gestational age over a 5-month period, which is a typical RSV season. And we saw similar efficacy across a number of different parameters that we looked at, including subtypes and disease of different severity. And this efficacy was consistent. One of the important things to note is really what happens with healthcare resource utilization with infants who have serious RSV disease. And it was really important for us in the context of our studies to look at that. The data I'm showing here is not a pooled analysis, but just wanted to highlight this data that came actually from our MELODY study that was conducted. And you can see here that admissions to hospitals, we favor efficacy with Nirsevimab. We also saw a reduction in admission to ICU, in use of CPAP or HFNC, which would be used for those infants who were admitted to hospital and needed to have either continuous positive airway pressure or high flow nasal cannula. Efficacy, infants affected, sorry, this isn't efficacy, this is infants affected by percent. Less infants requiring mechanical ventilation with Nirsevimab, and less use of supplemental oxygen in infants who received Nirsevimab during the course of the study. One of the other things that we looked at in the MELODY study was the outpatient visits and antibiotic use. And you can see on this slide that both of these things favored Nirsevimab and were lower than placebo. So we saw a reduction in outpatient visits for all-cause medically attended LRTI, as well as a reduction in antibiotic use in infants who received Nirsevimab compared to those who received placebo. In the study, we also looked at neutralizing antibody levels in infants who received Nirsevimab when compared to those who received placebo, and this is illustrated in this slide. And as expected, and this data was published in Nature Medicine, infants who received Nirsevimab, and we have data here from both our phase 2b study as well as our MELODY study, and you can see that infants who received Nirsevimab had higher neutralizing antibody titers than those that were receiving placebo on the slide. We also here demonstrated those placebo infants who either had an RSV infection or did not have an RSV infection and what their neutralizing antibody levels look like compared to those infants who either had an infection or did not have an infection who received Nirsevimab. And you can see that Nirsevimab recipients at baseline, their levels were more than 50-fold higher and at day 151 and approximately 7-fold higher at day 361 compared to placebo. The higher neutralizing antibody levels in Nirsevimab infants were actually observed through day 361, which is an indication that Nirsevimab may have efficacy beyond the typical 5-month period that we looked at in the course of the clinical studies during the RSV season. In our clinical development program, another important aspect was determining efficacy in those most vulnerable infants who were eligible for Synagis through their first and second RSV season. So I mentioned the MEDLEY study, which we conducted, and in that study, we compared children receiving Nirsevimab to those receiving palivizumab, given that they were palivizumab eligible population. And again, the children who would be normally recommended to receive palivizumab are indicated on this slide. We also conducted an immunocompromised infant study in children less than 24 months of age. And that was important, again, because of the significant morbidity and mortality related to RSV disease in these infants. But given that these infants are eligible for palivizumab, we agreed with regulators that we would extrapolate efficacy in these infant populations based on them having PK serum exposures comparable to those observed in the MELODY study. So in the study where we were able to demonstrate efficacy in the phase 3 population. So this slide illustrates the study schema for the MEDLEY study, which was the comparator study with Nirsevimab versus palivizumab. Again, this study was a 2-to-1 randomization, and you can see that the infants in the first RSV season received either the 50 or 100 milligram dose, depending on their weight band. So that is the less than 5 kg or the greater than or equal to 5 kg, or palivizumab, which is a 15 mg per kg monthly dosing regimen for a period of 5 months. And the arms of the study are here. We had the preterm infant arm and the CHD-CLD arm. These infants are typically recommended to have palivizumab in their second RSV season, at least the highest risk infants of these, and those will be the children with CHD or CLD. And so in that subgroup, we dosed children in a second season with a 200 mg dose of Nirsevimab or a dose or palivizumab. And we also looked at children who received palivizumab in their first season, but then Nirsevimab for their second RSV season, since we knew that that would be a case. Those cases would exist in the real world once the program was on the market. The design of the MUSIC study is illustrated on this slide. And again, this was a small study that was done globally, including 100 infants. This was an open label, uncontrolled single dose study. And that began later on in development once we had established the efficacy of Nirsevimab. And here you could see that there were 50 children enrolled for their first RSV season, receiving the dosing regimen that we described previously. They were followed through 361 days, and then children who received Nirsevimab for their second RSV season as a 200 mg dose. This study looked at safety, PK, and ADA assessments in these infants. On this slide, I just wanted to illustrate the serum concentrations over time in children and who were in the medley study. So that is the study that included the preterm infant population as well as CHD and CLD. And you can see here that their serum concentrations of Nirsevimab were high, looking very similar to what we saw for the healthy infants that we included in the phase 2b and phase 3 study. And then again, high levels of Nirsevimab in season 2 for the CLD and CHD infant populations. On this slide, I illustrate the efficacy extrapolation that we did to demonstrate that the children who were more vulnerable based on their disease status had similar exposures to those children who were enrolled in the MELODY study, greater than 35-week gestational age, healthy infant population. And you can see here the exposures in MEDLEY, very similar to and overlapping with those of the children who were enrolled in the MELODY study. We also looked at incidence of medically attended RSV LRTI in these infants through day 151. Again, this wasn't an efficacy study because if we had to do a non-inferiority efficacy study, that would require tens of thousands of infants and it just wasn't practical. But here you can see that we had really very few LRTIs in the children either receiving Pali or Nirsevimab. And we actually saw no LRTIs in the children who were enrolled in the study in the second RSV season. One of the things that we've looked at, and we've also published on this, is really what Nirsevimab concentrations look like compared to palivizumab. And here on this slide, you will see neutralizing antibodies of Nirsevimab compared to palivizumab in children, both in season 1 of the MEDLEY study and children season 2. And you can see that Nirsevimab has significantly higher neutralizing antibodies in these infants, is at much higher titers than the infants who received palivizumab. Nirsevimab has an has an acceptable safety profile. So this is really data from our clinical study program, and it's the safety profile through 360 days post-dose in both healthy preterm and term infants. And you can see a real balance between Nirsevimab and placebo groups with regards to AEs, adverse events of grade 3 severity or serious adverse events, treatment-related AEs, etc. I can say that the safety profile has held up since the rollout of Nirsevimab, and we know we've delivered millions of doses at this point to babies in the first and second seasons of launch that have occurred over the past year and a half. So just to remind everyone what the indication is for Nirsevimab, and it is for the prevention of RSV, lower respiratory tract disease, in neonates and infants born during or entering their first RSV season, and children up to 24 months of age who remain vulnerable to severe RSV disease through their second RSV season. And the presentations of both the 50 mg and 100 mg dose here for the weight banding strategy that ensures the right dose is given to the right infant is illustrated on this slide. Just wanted to highlight really the amount of work that was done to ensure that we had a pathway for approval, but also for recommendation of Nirsevimab in the US, and to ensure that this broad infant population could receive the monoclonal antibody. So this slide just illustrates some of the steps, and for many of you who may not be familiar with the steps to getting an approval and getting use, just wanted to highlight that. Of course, we had our BLA that was submitted. We had an advisory committee meeting for the FDA, the approval of the file, and then went to ACIP for the recommendation vote, as well as for the Vaccines for Children vote, which ensures that there is wide access for Nirsevimab in infants who would be covered by the public sector. We also had the publication of the ACIP recommendation in the MMWR, which is an essential step towards ensuring access. And then the first doses were supplied for that first season of launch in the US in September of 23, with the first immunizations occurring. As I mentioned before, implementation is very simple. So for infants in the US, for example, where there is a more winter circulation of RSV virus, those children who are born before the RSV season should get a dose at the beginning of the season, and they can do that in office or during a visit before a well visit before the RSV season. For those infants who are born in season, they can get it at birth before discharge in a hospital. And that is how implementation has been occurring. It is a simple vaccine-like implementation and provides protection to infants through the RSV season. It's an IM injection in a pre-filled syringe that's stored at 2 to 8. Here, I'm just showing the recommendations that they exist now for childhood immunizations in the US. And one of the things we're incredibly proud of is to see Nirsevimab as the first mAb on this immunization schedule, given how important it was to have a preventive intervention for infants. The real world impact of the intervention has been really staggering to see. And the first thing I would say, there's a lot of numbers in this slide from a number of different countries and many different designs of real world effectiveness studies on this slide, which I can't get into all of them. But suffice it to say, what we've seen is a real replication of the results that we saw in clinical studies with real-world effectiveness and showing significant impact against not just medically attended LRTI in the real-world setting, but also against hospitalizations with significant reductions in hospitalizations, ICU, and broad public health impact of the intervention in those countries that have rolled out all infant immunization programs. When we first launched the product, we didn't anticipate the response that we got in terms of how fast the uptake was. And so a lot of work was done to ensure that we had the supply that was needed for this RSV season that we are currently wrapping up. And that result in the US and actually globally involved a number of partners and a manufacturing footprint that would enable us to deliver the doses that were needed in the places that it was approved. And so just wanted to highlight here the ability of our manufacturing team to scale up to meet this demand, working very closely with Sanofi and our other partners, as well as the US government, LPPR, and the CDC to ensure that there would be a dose available for every infant who needed it in the US. And with that, I would like to stop and take any questions around the presentation.
Thank you very much. That was highly interesting and very informative. I really appreciate it. We do have a few questions already in the Q&A, which I can read to you.
Okay.
If anyone would like to ask a question verbally, all you have to do is raise your hand in the chat, and I can then unmute you and let you answer your question. So let me start with one of the questions in the Q&A. The question is about the Melody study. Have you measured neutralizing antibodies during the second season, in other words, course of this 365 days greater than the treatment? Does it maintain neutralizing antibodies?
So we have neutralizing antibody, and I think I showed that that slide through day 361, which for some infants would be going through to their second season, and what those levels look like, going compared to placebo. So I would say yes, they're higher than baseline, but we don't have a correlate, right, of protection for antibodies. So that's an observation, but no idea if that correlate is associated with a certain level of protection.
All right, thank you. Another quick question here is the current recommendation for this monoclonal antibody normal newborns also for women that have been immunized during pregnancy?
So for clarification, is the question, the Nirsevimab is indicated for use in infants entering their first RSV season. It is not indicated for pregnant women. Maybe I'm not clear on the question. Is it, can infants whose moms receive the maternal immunization also get Nirsevimab? Is that the question?
I think so, yes.
Okay, great. So actually, they don't need to get both, and the recommendations are very clear. If the pregnant woman gets vaccinated, and there's certain parameters around that when she gets vaccinated, etc., then there's no need to give the infant Nirsevimab. However, if certain things happen in pregnancy, for example, the infant is premature, or she has the delivery within a certain time frame after receiving her immunization, then the infant should get Nirsevimab.
Okay. Dr. Osterhaus is asking you, are there any indications of the induction of anti-idiotype antibodies?
No indication of that.
Okay. Another question in the chat is IgG FC mutated for longevity?
Yeah, so the YT modification is in the FCR region, and yeah, that binds to neonatal FCR.
Another question is, do you have any data yet on the performance of these monoclonals in low and middle income countries? Would it be affordable for them?
Yeah. So in terms of middle income countries, we have quite a bit of data as countries roll it out around the world. We also have data from South Africa, which was which was part of our clinical development program, and the antibody performs the same irrespective of settings. We don't have data in low income countries at this time. Our clinical development program was not conducted in low income countries. But there are ongoing discussions about how that could be done, and what infrastructure would be needed to allow that to.
So I'm going to allow Alex Richardson to ask you a question. So you can now speak.
Hello. I was wondering if you guys are planning on using the YTE technology on any other types of monoclonal antibodies for other diseases. Thank you.
Yes, the simple answer is yes. We use YTE in a number of the antibodies I mentioned before. So we've used it in COVID antibodies. We've used it also for some of our bacterial anti bacterial antibodies. So yes, we will use YTE across different pathogens, but we also have other extension half-life technologies as well.
Thank you.
All right, I think the last question we have in the chat is, what is the likelihood that you might see escape mutants emerging in response to using this treatment?
Yeah, so it's a very good question. And I didn't present on that today because that's a whole presentation in itself, but that's something that is that we continue to do is to monitor for potential resistance. So we did that in the context of the clinical trial. But we also do just general surveillance to see if there are any mutations that are popping up that in the binding site for Nirsevimab or anything that will impact the ability of the antibody to neutralize virus. And I would say those are rare, and we haven't seen anything pop up that actually becomes a circulating variant. We also have other people have published. There's an article from Moline et al. Published in JAMA that shows that independent groups have also looked in their effectiveness studies and sequenced escape any variants that they're seeing, and they haven't seen anything occur with regards to impact on Nirsevimab ability to neutralize circulating viruses. So I think, obviously something we will continue to monitor for, but we have not seen anything.
Okay, thank you. So I think that's all the time we have for the question and answer, and greatly appreciate you giving this presentation. It was highly interesting, and most people were highly engaged. So
Great.
thank you very much.
Thank you.
Just
Thank you so much.
remind everyone that this webinar will be on the ISV website. You can look at it there through YouTube if you need to go back and review this, or tell your friends who didn't get a chance to see it today, they can see it then. Until next month, take care. Thank you, everyone. Thank you.